Hey everyone, this is Dr.Mungli. In this video, I'm going to explain you different sources of energy sources for the brain and other tissues during fasting and starvation. Some of the processes that we are going to see today are glycogenolysis, that is breakdown of glycogen, gluconeogenesis, that is synthesis of new glucose molecule, ketone body synthesis and ketone body breakdown process, lipolysis, that is adipose tissue breakdown, and fatty acid oxidation. So these are the processes that we are going to look in today. So the plot I have already drawn here, and in the x-axis I have divided x-axis into hours, days and weeks. In hours I have divided that into 6 hour, 12 hour and then 24 hours, days from 2 days to 6 days and weeks from 2 weeks to 6 weeks. Now during fasting is basically, it is considered the 10 hours after meals is basically the fasting condition, 8 to 10 hours after meals, and then after 24 hours we call it as starvation. Now whenever we are in well-fed condition, means when we eat food, there will be release of insulin in our from the beta cells of pancreas, and the antagonistic hormone glucagon, it will be low because insulin has got an inhibitory effect on alpha cells of pancreas and decrease the release of glucagon. Say 2 to 3 hours after meals, what happens? During that time, insulin release from beta cells of pancreas decreases and the inhibitory effect on its alpha cells by the insulin is also decreased. It means the alpha cells will start synthesizing, start secreting glucagon. Glucagon appears in the blood now. So now the glucagon, what it does, glucagon, as you know, it is a hyperglycemic hormone. It is trying to bring up glucose. Now when the person gets into post-absorptive state, that is 2 to 3 hours after meals, glucagon predominates, insulin decreases. So what this glucagon does, what is the source of energy for the brain and red blood cells during this 2 to 3 hours after meals, means after in the post-absorptive state? So glucagon, first thing what it does is, especially in the liver, is going to break down glycogen present in the liver and release glucose 1-phosphate, and later it is converted to glucose. So glycogen breakdown occurs 2 to 3 hours after meals. So and it continues to go on and it's going to peak at 12 hours and then it will be totally depleted by 24 hours. Let me draw that curve for you now. So 2 to 3 hours, I'll take somewhere at this point. It rises. The glycogenolysis starts 2 to 3 hours after meals, that's a post-absorptive state. It reaches peak by 12 hours and by 24 hours it is completely depleted. This is what happens to glycogenolysis. This particular process is glycogenolysis, so which is mediated by glucagon hormone by activating glycogen breakdown enzymes, and they are glycogen phosphorylase and debranching enzyme. Now when the glycogen is completely depleted by 24 hours, now by this time brain has not yet adapted to use ketone bodies as the alternate energy source. Red blood cell, it is entirely dependent on glucose for its energy source. Now what are the other things that will help for the brain and red blood cells to survive, to get their energy source? During this time, another source for glucose is gluconeogenesis. Now gluconeogenesis is the synthesis of new glucose molecule from non-carbohydrate source. So the gluconeogenesis will be at a baseline at 2 to 3 hours after fasting, but it steams up at 12 hours and it peaks by 24 hours. When the glycogen breakdown is depleted completely, your gluconeogenesis peaks up. As you can see from the curve that I have drawn here in the red line, so it is peaking up by 24 hours and now it stays there for another 4 to 5 days or 6 days. Gluconeogenesis stays there at peak for another 3 to 6 days there, and then slowly it falls down. So during that peak levels of gluconeogenesis, what all the substrate for gluconeogenesis during that time? So the gluconeogenesis in the first week period it is predominantly supported by skeletal muscle proteolysis. You see the amount of protein that is present in our body is highest in skeletal muscle. Whenever person is in starvation, especially during first week, the skeletal muscle proteolysis will go on and it's going to release alanine and glutamine. And especially the alanine in the liver, it will be converted to glucose by gluconeogenesis. For every 2 alanines that are converted to glucose, you need 6 ATPs, and those 6 ATPs, they will come from beta oxidation of fatty acids. Soon I am going to explain you how exactly that goes on. Now lactate coming from anaerobic glycolysis will also support glucose formation. So for a 2 lactate converted to 1 glucose, you need 6 ATPs there. Glycerol coming from triglyceride breakdown, that can also be converted to glucose. Propionyl CoA coming from oxidation of odd number carbon fatty acids will also be can also be converted to glucose. So in the first 1 week time, gluconeogenesis is predominantly supported by alanine and partly supported by lactate, glycerol and propionyl CoA. But after 1 week, what happens? Gluconeogenesis falls, and by 2 weeks it just takes a constant phase. This is where the gluconeogenesis will be going on, and during the constant phase of gluconeogenesis, so basically it will be supported by lactate, glycerol and propionyl CoA. So as I have written here, gluconeogenesis during this time is supported by lactate, glycerol, propionyl CoA, because it is not and it is not coming from alanine, because by that time skeletal muscle proteolysis is already down, because the person want to save skeletal muscle, doesn't want to break it. Now the gluconeogenesis is an energy expenditure process. For every 2 alanine, every 2 lactate into 1 glucose, you need 6 ATPs in the liver. And who is going to give ATPs during this time? So under the influence of glucagon, 2 to 3 hours after meals, under the influence of glucagon, so there will be lipolysis going on in the adipose tissue. Adipose tissue start releasing fatty acid, start breaking triacylglycerol and releasing fatty acids. For 3 fatty acids are coming from 1 triacylglycerol, and also it's going to release glycerol molecule. So the fatty acids floods into the blood, they will be taken up by peripheral tissues, and peripheral tissues now start using fatty acids as their energy source, because glucose is less here. And the liver is going to use fatty acids, break the fatty acids into acetyl CoA by beta oxidation process. And the acetyl CoA initially diverted into TCA cycle, NADH plus H plus that comes out of beta oxidation and the TCA cycle, they will be diverted into electron transport chain and make ATPs, and these are the ATPs which will be used to run gluconeogenesis process. It means whenever there is a gluconeogenesis, beta oxidation of fatty acids will go hand in hand with gluconeogenesis. So let me draw now the fatty acid line here. So the fatty acid appears basically at the baseline down here 2 to 3 hours after meals, and slowly their concentration increases in the blood, because more and more fatty acid oxidation will be going on. So this line is for fatty acids appearing in the blood. So there will be constant amount of fatty acid that will be maintained after 1 or 2 weeks in the blood. So as the ketone bodies is like gluconeogenesis is going on, fatty acid is also going on, beta oxidation is continued there. Now what happens is, when there is too much of beta oxidation going on in the liver mitochondria, there will be so much of acetyl CoA that is elevated. So this acetyl CoA, it will be diverted into ketone body formation. So the acetyl CoAs are converted to ketone bodies, and now ketone bodies start appearing in the blood. So the ketone bodies are coming from acetyl CoAs of beta oxidation of fatty acids. So they will be at baseline. By 24 hours you start the significant amount of ketone bodies can be found in the blood. During this time, brain has started, it will start used to ketone bodies, so it will start utilizing ketone body for its energy needs, and by 3 days brain has adapted itself to use ketone bodies. During this time, skeletal muscle and other tissues will also start using ketone bodies along with the fatty acids. Only tissue that doesn't use ketone body here is the liver itself and also the red blood cell. Red blood cell do not use ketone bodies. Now, as the fatty acid appearance of fatty acids in the blood and also oxidation of beta oxidation of fatty acid goes on, ketone bodies will continue to rise. And as you can see here, ketone body levels rises and they peak by 2 to 3 weeks of time, so there will be high levels of ketone bodies. Now one thing, and during this time your brain has adapted to use ketone bodies predominantly, and it's going to save glucose for red blood cells. As you can note that brain starts to use ketone bodies at 24 hours after fasting. It has completely adapted to use ketone bodies by 3 days, and it is saving glucose for red blood cell. As the gluconeogenesis is falling, you can note that, take a note that gluconeogenesis falling. As the gluconeogenesis is falling, here, so there is increase in ketone bodies. So it means at this point in time, as you can see, there is glucose is falling down, ketone bodies are rising. During this time, your brain has adapted to use ketone bodies, and the glucose coming from gluconeogenesis, now it will be used by red blood cell. It will be spared by the red blood cell. And also, as the ketone bodies utilization by the brain is increased, your skeletal muscle will also decrease the use of ketone bodies for its own energy purpose. It will revert itself back to use of fatty acids for its energy purpose. So like this, during fasting condition and starvation, there will be adaptation mechanism going on in our body where different energy sources are used. So let me recap this now. So initially glycogen breakdown will go on and glucose is coming into the blood, and it will be used by the brain and other tissues for the energy purpose. So as the glycogen depletion is going on, gluconeogenesis will support the glucose needs of the brain and red blood cells specially, and this gluconeogenesis will stay there for at high levels for first week. After that it falls to a constant phase by 2 to 3 weeks, and it continues there. So during this time, when the glucose formation from gluconeogenesis decreases, now the brain has to search its energy source for something else. During this time, beta oxidation that is going on in the liver, so acetyl-CoA that is coming out of it is converted to ketone bodies. Ketone bodies appears in the blood by 24 hours. Brain has started to use ketone bodies by 24 hours. It has adapted itself to use ketone bodies almost completely by 3 days, and it is going to decrease its use of glucose coming from gluconeogenesis. And by 2 to 3 weeks, your brain has completely adapted to use ketone bodies. It is going to conserve glucose for red blood cells. So this is how adaptation mechanism will go on in our body. I hope you understood this particular concept. This video helped you to understand what all the energy sources of the brain and other tissues during fasting and starvation. If you have any question, so you can put the question down in the comment section below. So if you like the video, give thumbs up, and if for the regular updates on upcoming videos, you can subscribe to my channel. Thanks for watching again. See you next time.